Hello students and welcome again. So today we are going to discuss about the next topic of molecular biology, that is the packing of DNA into the chromosomes. So the most important functions of DNA is to carry the genes, the information that specifies all the proteins that make up an organism, including information about when and what type of cells and in what quantity of each protein to be made. So the genomes of eukaryotes are divided into chromosomes, and in this section we will see how genes are typically arranged on each chromosomes. So in addition, we will describe the specified DNA sequences that allows a chromosome to be accurately duplicated and passed on from one generation to the next. So in this slide you can see that is a prokaryotic packaging of DNA. So bacteria, we can say bacteria carry their genes on a single DNA molecules, which is usually double stranded, covalently closed and circular one. Okay, so this DNA is associated with the proteins those perform packaging and condensation of the DNA, but they are different from the proteins that performs these functions in eukaryotes. So although we can say they are often called the bacterial chromosome, it does not have the same structure of eukaryotic chromosomes. As you can see here, it is a bacterial DNA. Next, the packaging of eukaryotic DNA into a set of chromosomes. So in this there are levels of DNA condensation within eukaryotic chromosomes. So the first level of DNA condensation occurs when it is a 146 base pairs of the DNA which is wrapped around histone protein, so named as the nucleosomes. So DNA is wrapped twice around the histone core to form a beds on a strings levels of packing, termed as, we can say, as a chromatin. Okay, in the picture you can see. So this produces a fiber approximately about upon, we can say, 11 nm in the width. The second level of chromatin condensation is the arrangement of the nucleosome into a 30 nm chromatin fiber, where 6 nucleosomes per turn are found. So this structure is termed as a solenoid. Okay, and the next, the chromatin fiber is then arranged in the loop domain, which approximately 120 kb long. So this, we can say, these chromatin loops forms a rosette. So each rosette is we can say termed as a multi-loop sub compartments. Okay, so and comprises of 6 loops. So in a further 120 kb of DNA links each rosette. So the rosette is arranged at its center to the nuclear matrix and is associated with the histone 1. Okay, histone 1. So the final level of chromosome compactions occurs prior to the cell divisions in mitosis. The we can say mitotic chromosomes have a diameter of about, we can say, 700 nm. Okay, in the picture you can see. So the rosettes are the lined up and attached to a central chromosomes, we can say scaffold, and non-histone acidic proteins to form. So this is the packaging of eukaryotic DNA into a set of chromosomes. Next is the type of chromosomes. So when we say types of chromosomes, it will again divide into two parts: the number of on the basis of number of centromeres, and on the basis of position of centromeres of the chromosome. So on the basis of number of centromeres, the first one, that is a monocentric with the 1 centromere. Okay, dicentric with the 2 centromere, 1 in each chromatid. Okay, so the polycentric, we can say, with more than 2 centromeres. Acentric without centromere, it doesn't have centromere. So such chromosomes represents freshly broken segments of chromosomes which do not survive for long time. So diffused another, or we can say non-located centromeres is again, it is a type, are distributed throughout the length of chromosomes. So the microtubules of spindle fibers are attached to chromosomes arms at many points. So the diffused centromeres are found in insects, some algae, and in some group of plants also. So these are the types of chromosome on the basis of centromeres. Next, it is a type of chromosome on the basis of position of centromeres. The first that we can see is the telocentric, the last one. So it is a rod shaped chromosomes with centromeres occupies a terminal position. One arm is very long and other is absent, we can say. The next that is a acrocentric, second last. These are the rod shaped chromosome having subterminal centromeres. One arm is very long and the other is very small one. Next that is submetacentric. So these are we can say L or J shaped chromosomes with centromeres slightly away from the mid point so that the two arms are unequal one. Then the last that is a metacentric. So these are the V shaped chromosomes in which centromeres lies in the middle of chromosome so that the two arms are almost equal to each other. So these are the types of chromosomes on the basis of centromeres. So thank you very much.